Good afternoon. During these phenomenal 8 weeks in this beautiful city of Seattle, I was given the opportunity to work in Dr. Ferreira's lab with this incredible staff team. My presentation is on a novel mutation that was found in an aneurysm patient. As we all know, neuroscientists and neurosurgeons, intracranial aneurysms are both quite awful and quite common, with a prevalence of approximately 4%. Two kinds of aneurysm exist. The more common are known as saccular, while the latter, more rare, fusiform. Increasing evidence over the years has implicated a genetic role with regards to the etiology of intracranial aneurysms. Several genetic syndromes have become associated with brain aneurysms, but the role of somatic genetic alterations or mosaicism remains largely unknown. However, Dr. Ferreira's lab discovered that there is a somatic mutation involved, more specifically with the development of the less common fusiform aneurysms. These over here. Dr. Ferreira's lab recently published a paper about a particular patient, a 22-year-old man referred as JS. He presented at WMC with progressive exclusive right-sided fusiform aneurysms and hyperelasticity of the skin on his right side as well. It is worth mentioning that he actually first developed a fusiform aneurysm when he was only 9 years old. So it makes you think, what exactly is going on here with this patient? Why is he developing all these aneurysms 14 years later? Both his family history and all known connective tissue disorders were found to be negative. Genetic sequencing was then performed on both his blood and fibroblasts derived from the diseased areas, and further genetic testing from surgically extracted tissue revealed varying percentages of mutations. In other words, alternate allele frequencies. In particular, a single novel variant predicted to cause a change within the platelet-derived growth factor beta gene. Interestingly enough, mutations were only found within his diseased area, thereby confirming post-zygotic or somatic mosaicism. To briefly go over the gene of interest, activation of the PDGFRB receptor leads to the upregulation of the downstream signaling pathways, most notably angiogenesis. PDGFRB is normally expressed in several vascular cell types, such as parasites, endothelial cells, and fibroblasts. For further comparison, 6 fusiform aneurysm patients were genetically sequenced, and 4 of them were found to carry a PDGFRB mutation. For even further comparison, a total cohort of 32 saccular aneurysm patients were genotyped, and none were found to carry a PDGFRB mutation. Unfortunately, JS passed away over a year ago and donated his tissue samples to Dr. Ferreira's lab. The lab then proceeded to further uncover other areas where he exhibited alternate allele frequencies. And that's where I came in with my DNA extracting skills and extracted a total of 60 tissue samples. A solid phase extraction method was implemented specifically with the Mutation kit. This kit allowed the separation of DNA from proteins and other cellular components. Once these 60 samples were successfully extracted, some but not all were run through something known as droplet digital PCR, or ddPCR for short. This is a method that is based upon water, oil, emulsion droplet technology. How this works is by taking a sample and fractionating it into 20,000 droplets, and PCR amplification is conducted within each individual droplet. What is phenomenal about this technology is its ability to detect low-level mutations using low nucleic acid inputs. As it was aforementioned, only a few of these samples were run through ddPCR, therefore my graph is only providing just a glimpse of the alternate allele frequencies that were found. As you can see over here is the affected area, and as you're approaching towards his normal area, there aren't any allele frequencies. Thereby further confirming that there is somatic mutation involved. Mosaicism, sorry. And over here, this graph is just providing an overview of some, but not all, of the different aneurysms he developed and the alternate allele frequencies from these samples. And last but not least, these are the patient's skin and skin culture that were cultured in varying conditions, and when they were selected for fibroblasts, it had the highest allele frequency. Thereby, one could make the assumption that the fibroblasts within the artery tissue are contributing most to this patient's pathogenic phenotype. In conclusion, The samples were made to be run through the ddPCR and further analyzed. Additionally, a larger cohort of fusiform aneurysms will need to be genotyped for PDGFRB mutation. Lastly, treatments involving PDGFRB inhibitors and their efficacy will need to be assessed. However, in conclusion, the findings of JS-affected areas carrying varying allele frequencies are consistent with what the 2018 paper found, and finally, the cell of origin could be fibroblasts, thereby giving us hope for the future that we can find pharmaceutical interventions for patients such as JS. And this is definitely the most important slide from this presentation. I'd like to thank Dr. Ferreira and his lab for their ceaseless help and guidance. Any question I had, they answered with the utmost solicitude. Dr. and Mrs. Ellen Bogan for initiating this program 11 years ago and just doing everything in their power to further strengthen this program and have students from all over the country come in. And the entire UW Neurological Surgery Department for making us feel welcome. I honestly felt like an integral part of the UW Neurological Surgery Department. And everyone else that somehow, no matter how small or how large, made a contribution to this year's 2019 NSSB cohort. And obviously, thank you to everyone from the program for just being amazing. And that's our presentation.